Hello, everyone. Let me see here. So, a couple of things. One, your exam. Oh, wait, let me make sure that this is recording with. Uh, I think it's recording. It seems like it's recording. Anyway, so let me see here. Though it stopped sharing. Did it stop sharing? No, it's still sharing. God, am I this old? Jesus, man. All right, one, your exam is done. I need to grade your exam. You would imagine that Avenue would just have some like button I click that would automatically take all of the grades from the assessment and populate your could be grades on Avenue, but that's just not true. It doesn't have that. So um, I have to go take each individual grade and transfer it over, which I will do tomorrow. Um, I spent all morning in meetings, so I'm getting to this lecture kind of late today. Exam grade should go up on tomorrow. My office hours this forthcoming Friday will involve me going over each and every one of the exam questions, and you guys can ask why that answer was that answer and politely question whether or not that really is the real answer. And so you guys can do that as a swarm intelligence on Friday. I will host those office hours and send that invitation out on Zoom. So this week is supposed to be social insects, all of them. That's not possible. So in 3-ish hours, I'll probably spill over a little bit into subsequent lectures. I'll cover the general gist of social insect biology, broad brushstrokes of wasps, bees, and ants. I will not really get to termites because they're so different from everything else. But I might try to squeeze in something about termites, and then I will cover social spiders next week, and then whatever we cover next, I think it's like social reptiles and amphibians. So I think that's next. Anyway, so and then we'll have fish, et cetera, et cetera. So today we're going to go over the gist of social insect biology, the important themes that people consider in the literature, some misnomers in the literature that are echoed by intro to biology textbooks, and then we'll probably get to, as the lecture is called, wasps, but we're not going to get to that many wasps. We're not going to cover that much content that's specific to wasps at the end of this lecture. And I'll add to this lecture and update the version that's online as I produce the content. Okay, wasps. When you guys think of wasps, you probably think of like tiny little paper wasp nests with stinging flying insects, something like this yellow and black banded insect right here, right? But the word wasp, as we meet it today, covers everything from jewel wasps and hornets and paper wasps and yellow jackets and all of those, but it's a paraphyletic term, which means that a monophyletic term would be like the word wasp refers to everything that shares a common ancestor. So all the wasps share a common ancestor that goes back to about here, 0.3, right? That captures all the wasps, the jewel wasps, which are the most basal, and then the familiar wasps to you, including the little light pink one here, the Vespidae, which are what most of us think of when we think of wasps. But then all this other crap, like this gray box surrounds a lot of crap. And most of that isn't included in the term wasps. So like, for instance, right here, this yellow line, that's ants, all of them. And there are shitloads of ants. So like, when you use the word wasp, it's not a monophyletic term. We end up throwing out tons of other things that wasps gave rise to. Ants are just fancy wasps that lost their wings and became really, really social in the ground and in twigs and acorns and rock crevices and occasionally in leaf colonies. Anyway, we'll get to the various nest structures of ants later on. Bees, all those cool bees, halictid bees, sweat bees, stingless bees, honey bees, all of those, again, are like, they're just weird wasps, right? So the word wasp should encapsulate all those things, but people think of a very narrow chunk of what wasps are, and it excludes a lot of their descendants. So ants are fancy wasps, bees are fancy wasps, but we only focus on these sort of four groups out here, broad brushstrokes, when we think of wasps. And mostly when we think of wasps, we think of just one family, Vespidae. All right, so all of these eusocial insects, wasps, and their descendants, which are also modified wasps, ants, and bees, are all haplodiploid. Haplodiploid, it's a word you probably heard a lot, but it's a really weird mating system. So what it means is females are diploid. Just like you and I, they have hypothetically, two alleles, that maximum, for a specific locus. So they basically have two pairs of each of their somatic chromosomes, their non-sex chromosomes. So when a female Hymenoptera, a queen, or if she's not that social, just a female, when she produces gametes, it undergoes meiosis. So gametes only have one variant of each gene represented in the genome. So there's only one copy of each of these somatic chromosomes, non-sex chromosomes. And there can be crossover during those events. So basically her genome gets mixed up, and a subset of her gene by a crossover, one of her two copies of her genes will go into a gamete, which means she can produce gametes that, because of crossover, are incredibly diverse, well mixed, and will only have a subset of her total genes. Boys, i.e. drones in Hymenoptera, social insects, bees, ants, wasps, they don't have two different versions of every genome. They are haploid, like your egg cells or sperm cells, if you have those, are haploid. So, but the adult male, that drone that flies around, only has one version of each of its genes maximum. So when a father produces a gamete, it doesn't undergo crossover. It doesn't have to have, there's no coin toss about which version of that gene you get, because you've only got one. So each gamete's the effing same. So when a female mates with this male, she produces daughters. So following these lines over to the right here, daughters, the female symbol here, daughters here. Her daughters can have any possible combination of her genes. So she can get lots of different, she can get one of the two versions of her mother's genes at various loci, and it's well mixed because of crossover. So half of their genome is well mixed, just like you and I. Although your brother or sister, if you have one of them, share many of their genomes in common with you, there's a chance that they have different versions from their mother, different versions of some of these alleles from their mother or father. So you're not precisely the exact same mixture as to each other. And that because both your mother and your father are diploid, that mixing process occurs to produce each of their gametes, which means you and your brother and sister can actually be quite genetically dissimilar from each other. On average, you share 50% of your genes in common by common descent, but it's still 50%, it's just might be totally unlike each other. So that's why, one of the reasons why brothers and sisters are so dissimilar to each other. So we hit that process coming from mothers, meiosis, just like your meiosis, and that half of those daughters' genomes are well mixed like yours and mine. However, the paternal contribution from their father is clonal, which means that other half of their genome is just set. It doesn't vary at all. They get that paternal contribution, and as long as their mother's monogamous, every single worker inside of that society and every single queen that society produces is going to share 50% of their genome 100% in common. No crossover, no weird meiotic event, because meiosis doesn't happen here. That doesn't have to happen. They don't have to produce a number of chromosomes. And then the other half of their genome, just like you and I, they'll share 50% of their genes in common. What does that mean? Well, if 50% of their genome they have 100% in common, 0.5, and 50% of their genome they have half in common, like you and I, then that will create a 3/4 relatedness between daughters. So each worker inside of this society is a female, and so the other females bustling around her, as long as they all have the same father and the mother's monogamous, they're going to share 3/4 of their genome in common. When those mothers produce daughters, if they were to be able to produce those daughters, they would only share 50% of their genes in common, just like you are, like a normal mother and daughter or normal father and son in a diploid organism like you and I. So this turns out to be important because it benefits from Inclusive Fitness, at least hypothetically, for daughters in such a system to help rear additional daughters, because those daughters will have 3/4 of their genome in common, whereas their own should help rear their sisters, which will have 3/4 of their genome in common. If, however, they could by some chance event produce their own daughters, they'd only have 50% of their genome in common. So it pays more to invest an equal amount of resources into rearing sisters, because you get a 25% bonus on each and every single one of those individuals from Inclusive Fitness by rearing them and not your own offspring. Now, it's important to note that, like, rearing another worker that's sterile like you, like, even if you're 3/4 or if you were 1/2 related to her, like, another worker is just, like, not that big of a deal because they're not going to reproduce. Assisting a fellow worker doesn't increase your fitness in a direct way. What you really mean to say is helping rear additional queens who happen to be your sisters. The vast majority of females that these queens produce, your mother, let's pretend our mother is the queen, our mother is the queen, the vast majority of offspring that she produces are going to be workers like you and I. But later on in the season, or later on in this colony's development, she's going to start spewing out daughters that will turn into queens. So the real time that our Inclusive Fitness benefit could pay off is when we are rearing sisters of ours that we share 3/4 of our genome in common with that are going to become queens, because those are the reproductives. Otherwise, we're basically just helping build somatic asexual components of the colony, like you and I, workers that don't have direct reproductive output under normal circumstances. So this is in your Intro to Biology textbook, that this is the special sauce that made eusocial insects what they are today, why they evolved eusociality at least 4 times. Huzzah. It's in your textbook, Intro to Biology textbook, right now. It's probably wrong. Begins to fall apart. All right, so remember how I was qualifying everything, like if your mother uses just one father? On the left-hand side here is what happens when a mother who is diploid produces eggs that she's 50% related to, and those eggs can fuse. In these cases, she's having multiple daughters, and there's only one father who has no meiosis, and that half of his genome is always clonal. So, like I did the math last time, 1/2 of these daughters' genome is going to be clonal from their father, their shared father, and 1/2 of this genome is going to be from their mother, and that's going to produce 1/2 that genome is going to share 1/2 of its genes, so that's going to produce 1/4 relatedness. And if you sum that together, that makes 3/4 relatedness between daughters that share the same father. Problem: in lots of eusocial insects, their mom, she's not going to be so Puritan. In lots of social insects, this mother has to mate one time in her whole life in a mating flight. So the queens get released from the colonies, the males get released from the colonies, all the colonies are releasing their reproductives around the same time in synchrony, so that they don't just mate with males from their own colony. And most of those females that fly around, they're going to get mated by not one male, but by 3 males or 6 males or 29 males. And they're going to be inseminated out the wazoo. And then these females are going to store that sperm for the rest of their lives. Most of these queens, if they're an ant, land on the ground or begin to produce a cone if they're an ant, a bee, or a paper nest if they're a paper wasp, and they're never going to mate with a male ever again. They're certainly not going to mate with their sons, and they're going to use that sperm from that one mating flight for the rest of their lives. And in the case of some ants, all those fathers can be used, so all these insects can use these different fathers to rear different lines that might be good at different sorts of things, or to imbue their societies with genetic diversity, which is advantageous for reasons that we'll get into in just a second. But I want you to marvel at the idea that some of these queens live 30, 40, 50 years, and they only ever mated one time in their whole life to possibly a bunch of males for a day or two, and then they had to go excavate a colony, live in the bottom of that colony for the rest of their days, and they store sperm and keep it viable for decades inside of their reproductive tract. Amazing. We need special cryo chambers to keep human sperm viable for any significant amount of time, and it involves medical intervention to the extreme to keep that sperm viable, realistically, and technology. But these queens just have it just sitting around inside of them, and it's good that whole time. And while in a human situation, one sperm fuses with an egg, and we have millions of sperm, where only one will end up fusing with an egg, otherwise the egg is spontaneously aborted. In these social insects, it turns out that if you just count the amount of sperm that these queens have inside of them, they could run out of sperm because they're going to produce dozens, hundreds, thousands, sometimes millions of babies. And they've got that one mating flight from way back when that gives them all the sperm they're ever going to have. So why did I get on this diatribe? One, queens are good at keeping sperm active and alive inside of their gut, it's the reproductive tracts, not their gut tracts, Jesus, the reproductive tracts for a long period of time. No medical intervention necessary. Two, they're not Puritan. They need to get as much sperm as they can because they're going to be living with just that amount of sperm for the rest of their lives, and they better hold on to it. Anyway, so what happens when your mother has two fathers? She mates with two males. Or God, what if she had 29 different fathers, right? So like under those circumstances, you're no longer 3/4 related to all of your sisters. You're 3/4 related to all of your sisters that have the same father as you, in the left-hand panel here. But if not, if you have a different father, well, that means half of your genome is assured to be not overlapping. You have the white father, not the blue father. You're the daughter on the right. Mom used a different portion of her personal sperm bank to fertilize you and used a different one for me. I'm the blue one on the left-hand side. So we still share 1/2 of our genes from 1/2 of our genome in common, i.e. we have 1/4 of our genes in common. But then the other half of our genome, nope, they don't share any of that in common, or at least unlikely, unless those males are really closely related, which is not usually how it goes. So once you have mothers, these queens mating with multiple males, that benefit, that kickback of having 3/4 relatedness between sterile workers and their soon-to-be queen sisters one day, that goes away because a lot of those queens that they're rearing, a lot of those other workers that they're rearing only share 1/4 of their genome in common with them, and that's no greater in terms of relatedness than half sibs in human societies. If you have half siblings that share a mother or share a father, you share 1/4 of your genome in common. Half of your genomes come from each of those parents, and only 1/2 of those should you share in common because of meiosis and crossover. So you're back to just normal situations with polyandry. So monogamy, one male, one female, mate, monogamy. This is, I'm bearing in mind, like, to my knowledge, there are no known homosexual social insects that are obligate. So, we're talking about like general broad brushstrokes here, but there probably are exceptions to this. One male, one female, mated together, everyone's got the same mother and father, monogamy, genetic monogamy, genetic monogamy. Polyandry, many males, that's what that means. Poly, many, andry, andros, male. So like, yep, that female got mated with many males, probably two, at least two, possibly a lot more than that. And that's good from her perspective because she's getting a nice diverse genetic stock that she can use to fertilize her workers. Just in the case that one of the males that she mates with is a loser, or half of them are a loser, she's got an insurance policy against that because she mated a lot. I have to be careful here. Okay, so one, many queens mate multiply, which means that average relatedness from being 3/4, in real life, that's not true. It goes down. Daughters only share 1/4 of their genome in common with male offspring. So how does a male offspring form? They don't have a father. So just knock this father right out of this left-hand figure here. Father gone, blue gone. So the mother still undergoes meiosis and produces these eggs, and that egg will share, 1/2 of its genes in common with you from 1/2 of your genome, your maternal contribution. And so as a daughter, you're going to be rearing these eggs. If they're not fertilized, they develop into males. So mothers produce an egg, it isn't fertilized, she doesn't use a sperm to fertilize it, so it becomes a male. It doesn't just spontaneously abort, it just turns into a male. That's how males are formed, unfertilized eggs. Fathers don't produce males. Mothers produce males from not fertilizing their egg. So that means that that haploid cell, that haploid male, that you're going to still have to rear as a worker, doesn't have a father. You do have a father. That half of your genome you're not going to share in common with it because it doesn't have a father. And so it's only the maternal half of your genome that you could share in common with it, but because of meiosis and crossover, you're only going to share half those genomes half that genome in common. So 1/2 of 1/2 is 1/4, which means all those males you're rearing, You get gypped that way too. Haplodiploidy, my tush. Like you're like different fathers, and the fact that their mother is producing males screws you over time and time again because you're only going to share 1/4 of your genome in common with them. So the benefits, this like fantasy land that like you're going to rear all these queens and they're going to have 3/4 of the genome in common with you, malarkey, not true, whatever. No, it was never going to work. And so, but why is it that this haplodiploidy evolved? It's associated with the evolution of eusociality over and over and over. For a long time, most of the animals that we were aware of that had eusociality were haplodiploid. So it just seemed to reason that that would be the thing that gave rise to eusocial insects and eusociality in general. Nope. But now that we know a lot more about what's lurking around on planet Earth, there's a lot of eusocial animals that aren't haplodiploid. Naked mole rats, aphids, snapping shrimps, snails, like there are lots and lots and lots of eusocial organisms that are not haplodiploid. So that phylogenetic link, the idea that haplodiploidy was consistently giving rise to eusociality, fell apart as we discovered more and more taxa. So those are 3 giant holes in the hull of the battleship of haplodiploidy giving rise to eusociality. Now that doesn't mean that relatedness is unimportant. People have oftentimes said, well, if haplodiploidy is not important, if it's not the 3/4 genome hypothesis that gave rise to eusociality, then relatedness didn't matter. Non sequitur, not true, also not true. Relatedness still matters. So relatedness is important because the precursor to all of these polyandrous species, there are many polyandrous species of eusocial insects where females mate with many, many males, but their ancestor was not polyandrous. If you look across the tree of life for eusocial insects and you try to figure out statistically what the most likely common ancestor was to eusocial insects, the ones that have obligate eusociality, they become a worker or queen based on development and they can't reverse it. Obligate eusociality, cannot turn back. It is always a monogamous ancestor. So if you map that back onto the tree of life, you'll always see that each of those instances is more likely than not that each of those instances will have a monogamous colony where females only mated with 1 male, the left-hand panel here. So even though they're still rearing sons, and so the average relatedness is still 1/2, or it's still, at least the relatedness is 1/2 between the workers and not even lower because of having different fathers. So queen monogamy is the precursor to all known obligate eusocial insects. If you ask a eusocial insect biologist which taxa are eusocial and which ones aren't, no one can agree. No one can get over that. But there are at least 4 origins of obligate eusociality in the contemporary social insects. At least 2 in bees, at least 1 in ants, and then at least once in termites. Many people would say that no wasps are actually eusocial. So like, anyway, I'm not going to test you on that topic, but just know that people like to banter that about. Polyandry, that tendency for females to mate multiple times and have a little genetic diverse sperm bank or a little sperm portfolio, that only evolves later. Mind you, lots of species do it because it's advantageous, but it's only something that happens after the evolution of eusociality. So monogamy, monogamous queen gives rise to eusociality, and once that system is locked in, then they can evolve polyandry. They don't start off being polyandrous. So it still suggests, and there are polyandrous taxa that just don't ever give rise to eusocial insects, obligate eusocial insects. So that provides evidence, and that actually is, at least to some degree, helpful in facilitating the evolution of eusociality in bees, ants, and termites. Obligate genetic monogamy in the queens as the precursor to eusociality. Polyandry evolves repeatedly subsequently, and this assures reasonably high relatedness between workers, sisters, and so that's that. So they don't have like they have 3/4 of their genome in common with their female brethren, 1/4 of their genome in common with their male brethren, and on average it's 1/2 and not lower, because it could be lower if their mothers had been promiscuous. All right. And this will be the final topic that I talk about today because I wanted to keep this lecture kind of short. So evolving polyandry later is still quite advantageous. So why do they get this evolution of polyandry over and over and over? Why do monogamous, fidelity, Puritan social insects give rise to like floozy queens? Because it's really quite good to have diverse genetic stock inside the colony. So yes, having decreased relatedness means that it's easier to get things like cheating to evolve and greater conflict within colonies if there are multiple queens and things like that, right? But having increased genetic diversity increases division of labor inside the societies, that different lineages might be good at different things, kinds of tasks that a colony has to accomplish. And consistent with that hypothesis, if you look at queens that are mated multiply to many males or queens that are monogamous, there are higher levels of task specialization and higher levels of division of labor inside of societies that have more diverse genetic portfolios, those polyandrous queens and their daughter societies, relative to a monogamous queen and her descendant society. So there's greater levels of division of labor inside of genetically diverse colonies, possibly because different genetic lineages produced by males are better at different kinds of tasks. Second, there's decreased infection rates. Colonies that have multiply mated queens produce daughter societies that are less susceptible to various kinds of infections. This has been demonstrated time and again in domesticated honeybees, where we spend like 15 billion, I guess they produce about $15 billion a year in resources. So it's important that we keep these colonies alive. And what do you know? Genetic diversity is really good. And so one of the ways that you can ensure genetic diversity within the society is by making sure that your queens are being mated with lots and lots of males. We figured this out long ago. And then secondarily, once the colonies are, many colonies of honeybees die over the winter because they don't have enough resources. Colonies that have high genetic, this third thing down should not say high genetic relatedness begets increased overwinter survival. That's not true. It's the exact opposite. High genetic diversity begets increased overwinter survival. So genetically diverse societies are more likely to overwinter successfully and not perish at a critical time of year. So high relatedness favors division of labor. High relatedness decreases infection rates, and high relatedness increases survivorship of colonies over the winter. And although we know these outcomes best in honeybees, they're probably true in lots of taxa. And again, think about this as like having a diverse stock portfolio or having many different sires to use as possible fathers in your offspring. It acts as an insurance policy because you don't know what the future is going to bring, what kind of diseases are going to strike you down, or what kind of climate is going to be present for the next year or 30 years if you're in a long-lived insect society. So like, you need to buffer yourself against what could be a diverse set of future conditions. And the best way for you to do that, mate with many males, not just one. All right, and at that, I will let you guys go. I will stop my sharing, and I hope that this worked. I'll see you guys later on. Take it easy. Have a good one. Bye.